Thank you. So, hello everyone. I'm Hyeong Jun Jang at the Grade School of AI for Math. And then today I'm gonna talk about higher order Michaelis-Menten equations, the loss inference of hidden kinetic parameters, and then this is published in Nature Communication in 2025. So let's first start reading abstract. We So is everyone finished reading? So could anyone find some gap sentence for this abstract? It's here. The gap sentence is that yet the information provided by these parameters is incomplete and does not allow access to key observed such as lifetime of the enzyme substrate concentrate and then so on. So and then I think this abstract has some lots of specified terms for these biochemical reactions. So if you guys are not familiar with these words, then maybe this abstract makes it difficult to find some backgrounds or some important points of this abstract. So does anyone have another opinion of this abstract? If no, then I will explain each sentences and background for these papers. So first, I'm gonna explain about enzymes first. So enzymes are proteins involved in most of chemical reactions in our bodies. Actually, enzyme accelerates the speed of the biochemical reactions in our body by lowering the activation energies that needs to progress the reactions. So actually, it plays various roles in our body, such as digestions or immunity or liver and so on. So it plays it's very key proteins in our bodies. So how does this enzyme work? So this enzyme works by binding with substrates in our body and then metabolizing them. So here, the term substrates means that the materials or molecules that bind to the enzyme. So it contains lots of chemicals such as so endogenous substrates like hormones or drugs that we intake, or some environmental chemicals that we does not, such as pesticide that we does not intake, but we are exposed. So these chemicals can bind to enzymes and then metabolize. So you can just think these substrates as something like drugs or hormones and then so on. So the substrate binds to this enzyme, which I say E, and then make them complex. The complex can be go to in reversible way, so that it can go back to the substrate and then the E enzyme. And then finally, it becomes another chemicals called product P. So this is just some like conventional description of the enzyme reactions. So it contains binding and then dissociation and catalysis. And here, as you can see, there are some parameters of the enzyme reactions, like these values. So here K on means the binding rate between E and S, so that it becomes to ES. So it means how fast this binding occurs. And here K off means how fast this dissociation occurs. And then here K cat means how fast this formation or catalysis of the product from this ES occurs. Then what is the relationship between this K on, K cat, K off, and then the variables like substrate concentration, enzyme concentration, ES or P, and so on. So to describe this relationship, the Michaelis-Menten equation is proposed, and then this describes how the enzyme occurs under certain parameter values. So this is the enzyme reaction that I showed before. And here, Michaelis-Menten equation, which is proposed in 1913, says that it says the relationship between the initial turnover rate V0 and then the substrate. So what is the initial turnover rate? So it is written mathematically in this form, and it just means that the initial velocity that this product formed. So you can just understand that this V0 is some like the speed of this product formation. And then here, because Menten looks like this, so K cat times ET times S over Km plus S. So here, ET is just something like initial enzyme concentration that we give at the start of the experiment. And then if we draw this, the relationship between the V0 and then the S, then it looks like these hyperbolic curves. So as the substrate concentration increase, the formation speed of these products increase, but it does not increase infinitely, and it becomes saturated. And then here, the maximal velocity of this formation is described by this K cat times ET. And then here, you can see that there is something like Km that makes some hard maximal velocities. And this Km is defined by this K off plus K cat over K on, and then this is called by Michaelis-Menten constant. So what this Michaelis-Menten constant means that if this Km becomes large, that means this K on becomes small. So this binding rate becomes low. So it means that the affinity between this substrate and then enzyme is low. And then vice versa, if Km is low, then we can say that the affinity between this substrate and then enzyme is high. So as you can see, these parameter values, K cat and then the Km, represent some properties of the enzyme reactions. Then how can you obtain,
Is this Michaelis-Menten equation the same as the single molecule Michaelis-Menten equation in the abstract?
No, actually different.
Oh, I see. Okay.
This is something like conventional versions.
And then there is another thing.
Yeah, right.
The single one.
Yeah. Well, thank you. So then how can we obtain this K cat and Km value that describes the property of the enzyme reactions from the experiment? So actually this MM equation, because with the equation, enables to estimate the K cat and Km from the experiments by using the simple linear regressions. So let's see the form of the Michaelis-Menten equation again. If we just flip over this the left-hand side and the right-hand side, then we can make these double reciprocal equations. And then as you can see, it has some linear relationship between this 1 over V0 and then 1 over S. So by simply applying the linear regression, we can get K cat and Km from the data. So I will explain the details. So first, let's do experiments with several S values and then fixed enzyme concentrations. Then we can get these data from several substrate values. And then by fitting the linear model, then we can get the slope and then the intercept, which is described as like this from these double reciprocal equations. And then by simply rearranging these A1 and B1, which is derived from the experiments, then we can compute the Kcat and Km values like this. So as you can see, we can get this Kcat and Km values from the express by using the Michaelis-Menten equations. And then actually, these Kcat and Km values are very powerful to predict the dynamics of the drugs in our whole bodies. So this Kcat and Km values actually enable us to predict the whole body dynamics of the drugs. So there are some like famous model called physiologically based pharmacokinetics model called PBPK model. And then it describes the whole organs in our bodies and the blood vessels so that it predicts how the drugs moves and then metabolize in each organs through this and then moves through the vessels. And then here in the liver part and then the gut part, the enzyme goes as a metabolism. So the enzyme reaction occurs in liver and intestines. And to describe these enzyme reactions, actually the Michaelis-Menten equation has been used. So by simply putting these Kcat and Km values from the values that we got from experiments, actually we can describe this whole body pharmacokinetics. So actually it looks enough that to estimate Kcat and Km values, it looks enough to describe the dynamics of the drugs in our bodies. But actually, is it true that only this Kcat and Km are sufficient? And then the answer is no. So the hidden kinetic parameters that we cannot obtain from Michaelis-Menten equations are necessary to understand the enzyme reactions. So let's see these enzyme reactions again. And then from the parameters that we can obtain from Michaelis-Menten equation is Kcat and Km values. But we cannot get information of these K-off values and the K-on values from the Michaelis-Menten equations. We can only get these compressed values, compressed informations. And knowing Km is not sufficient for understanding and utilizing enzymes and drugs. So the first enzyme reactions having a different properties actually can show some equal Km values or similar Km values. So let's say we have some fixed K2 values as 0.01 and then ET values as 1. And then first, let's say K-on value is 100 and then K-off value is also 100. It means that this binding and unbinding occurs very fast. So fast binding and fast dissociation. And second, let's think this another case where K-on and K-off values are very small, which means that this binding and unbinding are very slow. But if we see, so although these reactions have some two different properties, if we see this Km values, if we calculate these Km values, then their values are very similar. So from this, we can say that Km serves only as compressive information, not this detailed information of the each state of change of the enzyme kinetics. And second, knowing these hidden kinetic parameters Kcat and K-on and K-off actually enables some precise drug design. So in the pharmacology, there are some important notions, people like drug resistance time tau, and it means that how long the drug stays at the target that we want to target. And let's see some graph like this. So it describes as time goes on how the drug concentration change in our body, and then how the drug effect change in our body. So here, the gray graph is the change of the concentration of the drugs in our body that can be predicted by the PBPK model that I explained before, and then actually this PBPK model can be described only by this Kcat and Km. So as time goes on, by the metabolism of the enzyme, the drug concentration decreases. And then let's see these two different cases. Actually, these two different cases have some the same kinetics of the drug concentrations. Then let's see, we have some long time drug residence time, and then we have some very short time drug residence time. And as you can see, if the drug residence time is very long, then the drug effect is almost constant as although the time goes and goes over the 24 hours. But however, if the drug residence time is very short, then the drug effect steeply decreases.
I'm sorry, is that the drug, the substrate,
Yeah.
and the tau, the drug residence time tau is related to those parameters, or is it something just totally independent?
Actually, this depends on these case of parameters. So as you can see, depending on this residence time, the drug effect differs. And then as Hyeong said, this drug residence time is dependent on this K-of values, which is hidden.
Is it? Is that the target is the enzyme?
Target is enzyme.
Because here, for me, that tau should be how long that the substrate binds to the target,
Yes.
and the K off is corresponds binding to the enzyme. So
Yeah,
I
yeah.
think your explanation about that when the situation that enzyme is the target,
Yeah.
is it right?
Yeah, that's right.
Well, one minor comment on that is, if that was the case, then tau would be 1 over k off plus k cap.
I think that might be appropriate to describe these kinetics.
Or some like partial time scale
Mm.
can be used to describe the only binding and unbinding,
If we only focus on unbinding, then
preserving
describe
the
it
substrate as
correctly.
S, not product P.
yeah. But if we contain also these values, then maybe we should add K-cat. But although we add K-cat, but we should need information for the K-cat. So
And then we will be using this tau like many times later, right?
no, actually. Actually,
And
yeah,
in this paper.
yeah.
Okay, then, well, we'll see.
Okay. So what I can say that we can evaluate and modulate how, and thus we can modulate drug effect if we can access this K-of values. But because the Menten's equations cannot do it, so therefore this study presents something called a high-order Michaelis-Menten model that can accurately estimate the hidden kinetic parameters. So the conventional approach with Michaelis-Menten equation actually do the turnover rate experiments that we showed before, plus Michaelis-Menten equation, so we get K-cat and Kn values. But this proposed approach actually used the single molecule experiments that we showed in the abstract, plus they utilize higher moments. So they can get K-cat, K-on, and K-off values from the experimental data. So first, let's go through what these single molecular experiments is. So this study focused single molecular experiments to get more information about the enzyme reactions, and to enhance your understanding, I just brought some similar cases to enzyme reactions. So let's imagine that we want to measure some sleep onset under a certain caffeine concentration before we go to sleep in a dormitory of the students. So if we take low caffeine concentration, then they can get very good sleep quantities, so they can wake up very well. So here yellow means wake up, and then the black means sleeping. But if we give very high concentration, then they will have some very poor sleeping conditions, so that they cannot wake up very well. Then how can we measure this timing from sleep onset, from sleeping to this waking? So how can we measure this sleep offsets? There are some two ways to measure that we can think. So the first thing is just let's measure some electricity monitoring of the entire dormitories. So if they wake up, then they will turn on the light so that the power of the use of the electricity becomes increases. And then let's just measure the timing when this uses of the electricity goes over the 1% of the whole usages. And then the second approach is let's just give the Galaxy Watch to all students so that we can measure the wake up time of each student. So in these two approaches, the first one is very simple. We just see how this electricity usage changes, and then it's very cheap. But it neglects the individual differences. It only measures the whole actions of the students. However, if we give Galaxy Watch, then it's very expensive. However, we can measure some individual difference of the wake up time. And we can think these schemes similar to the enzyme reactions, and then the timing that what we want to measure in the enzyme reaction is the turnover time. The turnover time is a timing where the substrate change into the product in a single enzyme. So if the substrate is low, then the takeover of the enzyme will be very low. But if the substrate becomes high, then the takeover of the enzyme will be very high, so that the formation of the product will be very active. Now let's think similar. So in the conventional approach, like the bulk experiment, we usually measure the timing when these product concentrations goes over 1% in a whole plate, so that we can measure something like the turnover rate, how fast this the system makes the product fast. However, in the single molecular experiment, actually is similar to that, to giving some Galaxy Watch to each single enzymes. So it can measure the timing when the substrate changes the product for each molecule, so each enzymes, so that we can measure the turnover time of each enzyme. Also, we can obtain some distribution of the turnover time t-turn, so that turnover time t-turn from the single molecular experiment can give more information about enzyme reaction compared to these bulk experiments in the conventional approach.
So should be as relative, because one E can change several things.
Yes. Sorry.
So for me, it's not so clear that why you put E here. Because you correspond one value to each E.
Yes.
But each E, so after turnover, that the same you can do the same thing to another.
Yeah, well, we only focus on the first time that the substrate changed to the product.
First time. Then not all E is working.
Yeah, actually, we set as you can see in this, we set some interest regions for each enzyme. So we only focus on the single enzyme, but we can do it for the multiple enzymes, but we set the interest region separately.
We can fix the location of the enzyme.
Yeah, actually, they said it's possible from the 20 years ago. So
Maybe that's why the photo over there depicts small screen. connected to the enzyme or something. I don't know.
Yeah, okay, got it.
So in summary, we can measure the distribution of the turnover time by using the single molecular experiments. Then let's describe the enzyme reactions which fit to the single molecular experiments. And then they say that the single molecular experiments can be described by called something like linear approach. And then this linear approach requires the fewer assumptions to describe these enzyme reactions. So single molecular experiments, actually now we are interested in timing of the state changes of the individual enzymes. So in these enzyme reactions, what we are interested in is the time, like this T on, where this E plus S state change to this complex state, and then the first time that this ES changed to the E plus P, and then the first time where this ES changed to the E plus S. So we describe the first time of the state change at this T on, T cat, and T off. And then what we observe is the turnover time T turn. And by using these notions, we can actually describe the turnover time from E plus S to E plus T in a mathematical way. So let's see this state change E plus S to E plus P in a detail. So we can think the first case where the catalysis occurs faster than the dissociation, which is described by this T cat is lower than T up. Then let's see this graph. So the x-axis is the time, and the y-axis is the state, E plus S, ES, and then E plus P. So we first start E plus S state, and then after this T on time, it changed into the ES state. And then since the catalysis occurs faster than the dissociation, after this waiting time T cat, it becomes product. So by seeing this graph, we can say that the time takes from this E plus S state to E plus P state is T on plus T cat. So we can say that the turnover time T turn is T on plus T cat when the catalysis occurs faster than the dissociations. And in the opposite case, where the dissociation occurs faster than the catalysis, also we start from the E plus S state, and then after this T on, it becomes the ES state. And then since the dissociation occurs faster, after this T of time, it goes back to the initial state. And then we repeat the same process again. So now let's say that the catalysis occurs faster. Then after this T on time, it goes to the ES again. And then since in this event, since the catalysis occurs faster, so that it becomes product. So as you can see, here is another turnover time. So the whole turnover time, the whole time that takes from this E plus S state to the E plus P state is the T turn, and then it is the summation of the T on plus T off plus this whole T turn time. So by using these notions T on, T cat, and T off, we can actually describe the turnover time under these specified two conditions. Yeah.
So in the single molecule, in the single enzyme, always time there will have different random.
Yeah, actually these times are random. So it changes as time goes on.
Exponential distribution.
Actually, it does not require any distribution of the decay of the
But this methodology is not, yeah, it's not limited to, yeah, possible.
Actually, this description of the linear approach does not require any assumptions, but later this methodology work completely under the Markovian
Okay,
engine.
also maybe to calculate to calculate the expectation value of the.
Yeah.
So maybe the geometric distribution comes out because T of can refers to T can refers.
Mm. Yeah. Any other questions? Okay, then let's move on to another definition. So here is the T term. And then actually also interested in the waiting time, which is value that we cannot observe. So waiting time, WES, means the first time when this ES changes to another state, like change to E plus P or change to E plus S. So we can also think this change of ES to another state one by one. So let's first see the first case where this catalysis occurs faster than dissociation, and it's the graph that we see before. And then here, the time, waiting time, where this ES changes to another state is actually this Tcat. So it takes Tcat to change from ES to E plus P. So the waiting time under this condition is actually Tcat under this condition, Tcat lower than T off. And then we define this condition value as a WES cat. And also in the opposite case, we also have these graphs. And then here, the waiting time where this ES changes to another state ES is T off. So in this case, the waiting time when this Tcat, when this dissociation occurs faster than the catalysis is T off under this condition, and we define this variable as WES off. So the definition of the waiting time is for the description of the T turnover time laters. So actually, this linear, as Yungmin said, this linear approach requires no assumption for the T on waiting time and then the waiting times. So it describes the enzyme reactions in general, and actually it can describe some like multiple step reactions. So like let's say we have multiple steps to turn over from this ES to EP. But by just saying this whole time as a T, by this time as a T on, and then by saying this time as a T off, we can just simply describe these complex reactions into these simple diagrams. So we can describe the enzyme reactions in general. So in these linear approaches, actually quantities other than the turnover time are difficult to obtain. So here is the linear approach. And then this is the definition of the turnover time that I explained before. And actually, by using a single molecular experiment, we can measure the turnover time, distribution of the turnover time like this. And then here Ft tran is the density function of the T term, and then I will use something like F sub notations to describe the probability density functions. But however, the waiting times or these binding times, although we want to get this information of these waiting times and binding times, actually these processes occurs very fast. They change and then the reversible wave occurs very fast, so that we cannot actually observe or measure the change time in an experiment. So what this study want to go over is how to infer these informations from the the distribution of the turnover time. And then to infer these hidden quantities waiting times and then the binding times, higher moments of the distribution of the turnover time can be utilized. So let's say we have some observable t-turn distribution, and then our goal is to infer the hidden variables of the and then the distribution of these hidden variables. And then the approach that this paper takes is first calculate the moments of these distributions. So here this bracket means expectations, so that the bracket t-turn means the mean of the turnover time, and then we want to get this study get some higher moments like expectation of the t-turn square, expectation of the t-turn cube, or the expectation of the power of the n. And it's actually calculated from these distributions. And then they find some relationship between these observable moments and then the hidden variables. And then by solving, actually we can get the infinite orders of the moments in an infinite numbers of the moments. So by using this infinitely obtainable number of the relationship and then solving these equations, they want to infer the hidden variables from this relationship between the moments and then the hidden variables. So first, let's go over this, how to calculate the moments. And then they use Laplace transformation to get these moments. So actually Laplace transformation enables easy calculation of the moments. So let's say we have some functions defined on the positive and non-negative t. And then the Laplace transformation means that this f hat k, which is defined by this interval form, so if products with this exponential minus kt, and then take interval over this non-negative regions. And if we change this function to the probability density function of the random variable that has some positive range, and if we take the Laplace transformation, then it becomes simply, by definition, it becomes an expectation of these values, like exponential minus kt. So by taking, and then under some regularity condition where, let's say that we can change the order of the differentiation and then the expectation. And by simply assuming these conditions, if we take the first derivative to this if we take first derivative to this Laplace transformation, then the derivative goes into this side, and then it becomes like this minus the expectation of the minus t exponential minus kt. And if we also take the second derivative, then the derivative goes inside to this part, and then it becomes like t squared times this exponential. And then if we go nth derivative, then it becomes like this form where it contains something like t and t power n. And by just simply taking the by simply put the k equals 0, then we can get the first moment by just putting this k equals 0. And then we can also get the second moment by just putting this k equals to 0, and then so on. So actually we can get nth moment of these variables by just simply taking some derivative of the Laplace formation transformed the density functions. So now they apply this Laplace transformation to the density function of this t term. So all moments of the turnover time can be estimated and calculated through the Laplace transformation to this to the density function of the turnover time. So here is the definition of the turnover time, which is observable. And then there are some hidden variables like waiting times and the binding times. And then actually I don't, due to the time limit, I should omit the exact derivation of the Laplace transformation. So you can see the derivation in appendix 1, and I think it is not that difficult. So you can go through it if you are interested in. So by just using the definitions, we can actually derive the Laplace transformed density function of the turnover time like this. So as you can see, it is described like hidden by the hidden variable, and here the pi cat means the probability that the catalysis occurs faster than the dissociations. So it describes by these hidden variables. And then in this paper, they assume that this turnover time follows the exponential distribution like this, which means the memoryless binding. So they simplify this equation into this form. And what is the memoryless binding? So memoryless binding means that the turnover time follows the exponential distribution. And the meaning of that is as follows. So let's say this red curve is the density function of these exponential distributions. And then let's say that we get some history of this turnover time. So let's say that there was no binding until this time s. Then by conditioning this, we can describe the density function under this condition like this. So before Ts, the probability that this T on occurs is 0, but after that we have the probability distribution like this forms. So this means that we got this region describes under this history how this turnover time occurs over this s plus t. And if we bring the shaded region, it looks similar to the exponential distribution. And actually, under the condition history, it also follows the exponential distribution, which means that although we know the history, but the future events does not depend on the history that we know. So history does not affect to the future events. So this is the reason why this exponential distribution called something like memoryless binding. And then this exponential distribution has this Laplace transformation form like this. So by putting this into these regions, we can actually derive this right form. Then why does this memoryless binding make sense? Actually, in these cases, because enzyme and substrates are quite well mixed. So let's say in the T0 time, we have this well mixed condition where this enzyme are surrounded fully by this substrate. And then although the time goes so that the binding event doesn't occur, it's still well mixed between the substrate and then the enzyme. So that binding probability at this time and the binding probability at this time is actually same, so that it does not depend how long the binding probability does not depend how long binding not occurs. So you can understand this is like the similar scheme of like this. So let's say you are surrounded by some COVID patients, then the probability that you are infected at this time and the probability that you're infected from the time as the time goes to something like one hour later or 2 hours later will be same. So we can actually understand this scheme as like that, curvature. So we now finally get the Laplace transformation of the density function of the t turn, so that we can finally calculate the moments of this turnover time. And actually, the mean turnover time has a linear relationship with this 1 over substrate concentration. However, still, hidden kinetics parameters cannot be obtained. So let's first, from these Laplace transformations, let's first calculate the mean of the turnover time. So we can just simply calculate this by taking this derivative, and then it looks like this form. So let's say the slope as A1, and then the intercept as a B1. And then you can see that this turnover time, mean of the turnover time, and then the 1 over S has a linear relationship. And then it does not depend which distribution that we give to this T off and T cat. It always shows the linear relationship without concerning the distributions. But still, we cannot get individual hidden values. So we have some two equations about this A1, B1, which we can obtain from the experiments, and then the hidden variables like this. The first equations and then the second equation for each A1 and B1. However, the hidden values that we don't know is pi cat, k on, and this mean of the waiting time. So the number of the hidden values is more than the number of the equations that we can obtain. So actually, we cannot get the full information of the hidden variables. Then how about utilizing more than higher moments? Although we are using higher moments, when we use higher order moments, we can actually calculate the higher moments. However, the linear relationship that was shown in the mean turnover time is break up. So from this Laplace transformation, we can compute the second moment, third moment, or even this something like some combination of the first moment and the second moment. However, if you see this graph, this is the x-axis is the 1 over S. You can see this as 1 over S, and the y-axis is the second moment, third moment, and then the CV values. You can see that the moments and then this 1 over S does not show the linear relationships. So in these higher moments, we cannot get some kinetic information as we did before for the Michaelis-Menten kinetics, or did for the first moments, the mean of the turnover time. So to make the linear relationship, how to make this linear relationship even in the higher moments? So they did not focus on that each moment, but they want to find some combinations of the higher moments that show some linear relationship with this 1 over S values. So from this Laplace transformation, they just flip over these functions, and as you can see, if we just flip over, then these functions have some linear relationship with these 1 over S values. And then to get some higher moments, they use Taylor expansion to obtain the linear relationship for the all orders. So first they take Taylor expansions to these functions so that they can obtain this form. And then let's say this inner form as the omega n, and then take Taylor expansion for this part. And then let's take this part as an an, and then take Taylor expansion for this part, and then let's take this inner part of the summation as a dn. Then they also take the Taylor expansion to this one, and then this one can be described as follows, where this delta n0 means that delta n0 is equal to 1 if n is 0, and delta n becomes 0 when n is not 0. So by taking the Taylor expansion to each part of these equations, we can describe like this. So the summation over this omega kn is equal to summation of the series of this linear form of the combination of this an, bn, and this delta n0. And using the property of the power series, where this if the power series is equal, then the each components of the a and b n is equals for the all n's. So by using these properties, we can get this relationship for all order of n. So here, omega n is since this Laplace transformation derivative of the Laplace transformation can make the moment, we can say that this omega n is something like combinations of the higher moment. And then this an, bn are something like described by these hidden variables. So finally, we can get the relationship between these higher moments, combination of the higher moments, and the hidden variables. So through this, the equations for the higher moments that show some linear relationship can be obtained for the all orders. So let's see these tables. So here omega n is from this omega n, an, and bn are from the whole equation that we derived. So in the first order, as we can see, as we saw before, the omega n is just the mean of the turbo time, and then an looks like this, and bn looks like this, which is described as the hidden variables. And if we take the second, if we take the n equal to second, then it's also described like the combination of the first moment and then the second moment, and then the an and bn are described by the hidden variables. And if we take third orders, then this omega n is also described by the combination of the higher moments, the third moment and the second moment and the first moment, and then the an and bn are described by the hidden variables. So if we see the relationship between the 1 over s and then the omega 2, it shows linear relationship with dependent on which distribution we assumed. And also in the third order cases, it also shows the linear relationships. Then, by using this, can we infer the hidden variables from these higher order MM equations? Actually, higher order MM equation enables some complete finding of the hidden variables for the specifically for the Markovian enzyme. So this is the relationship where we can get from this omega 1. And let's say this first, the slope as a A1 and then the intercept as a E1. And then if we take the omega 2, then the moments, combination of moments looks like this. And then we can get the A2 and B2. And actually this A1, B1, A2, B2 can be inferred, can be derived by taking the linear regressions to this higher combination of the higher moments, which can be obtained by the experiments. So by rearranging the relationship between AN, BN, A1, B1, A2, B2, we can get the formation of the K1, formation of the K pi cat, formation of the waiting time, mean of the waiting time, the formation of the another mean of the waiting time. But as you can see, still there are some unknown values, like hidden values, so that we cannot obtain. So the hidden values cannot be obtained. But if we simply assume if we simply focus on the Markovian enzyme, then the whole problems that the hidden values is not obtainable is solved. So here, Markovian enzyme means that it takes so each time of the state change takes the exponential distribution. So this is called Markovian enzyme. And then actually Markovian enzyme has something like two important properties. So as I explained before, memoryless binding, memoryless dissociation, and memoryless catalysis can occur. And also, if we assume this Markovian enzyme, then actually this waiting time of the catalysis, waiting time of the dissociation, and then the waiting time of the state change of the ES has some same distribution, which means that the mean of the ES cat, WES cat, mean of the WES op, and the mean of the WES are equal. So by put this relationship into these each formulas, then we can simply describe this K1 pi cat and this waiting times looks like as this. And then since these blue variables are obtainable by the experiments, actually we can describe these hidden variables only the information that we can obtain from the experiments. So now we can obtain these hidden variables from the data. And then these results in the accurate and precise estimation of the hidden parameters for the microbial enzymes. So we have formulas like this, and then using these formulas, we can actually also describe the K off and K cat like this. So if we see this graph, so this graph is the y-axis is the ratio between the true values, the lower is the true values, and then the inferred values. So if its values is closer to the 1, then it means that the estimation result is very accurate. And then they actually did numerical experiments under these different numbers of enzymes. So they use 100 enzyme to this 10 power 6 enzymes. And as you can see, although we are using this 10,000 enzyme, the accuracy and then the precision of these estimation results are very good. So by only using the information from the turnover time, we can actually get the full information of these hidden variables. So it's accurate, precise, and identifiable beyond these compressed KM values.
Did they use the simulation data?
Yeah, they only use simulation data.
And then simulation comes from some stochastic model.
Yeah, right. I don't remember which distribution they assumed. Actually, this is microbial enzymes, so they all assumed the exponential distribution and they use the stochastic simulation.
They assume that S is a constant or
S, they varies the S. So to obtain this A1 and B1, A1 B1 by linear regression, they should vary the S.
I can see that they have to vary the S value. The initial conditions for
initial conditions for S values.
Throughout within the single simulation set is S variable.
They did experiments in the different acids, initial conditions of the acids.
So let's say we fixed the initial condition for S to get a single point.
Yeah.
And then we have to run the stochastic simulation.
Mm.
And then throughout the stochastic simulation, is S changing or? It depends on the type of the stochastic simulation setup they use. But it seems like all the derivation they have relies on the assumption that S is kind of because S represent the number of substrate that could interact with the single enzyme, right? With a specific physical distance.
But to see the change.
And technically it changes because enzyme literally changes one S to P. If S, like the number of S like 1000, it is not that important. 1999 is not that different. But when we consider the single molecule experiments, then there are not that many different like substrates nearby the single enzyme, and that can change the number of substrates like significantly. And in their derivation, But if that change, then that actually could alter all the derivations. If that is not clear, that's fine.
Actually, I didn't check it while I'm reading this paper, so I should check.
But we should keep in mind
Yeah.
the equations that we have is assuming S is kind of the constant. I'm not sure if this
Here-
is like
here- The S means actually I should make notation more carefully, but where is it? So here, this S is the initial condition. So this S does not change. But while they are doing some experiments, maybe they let S changes as the time goes on by the state
Yeah,
changes.
for the stochastic simulation, I think so.
Yeah.
And then
But this S is the fixed value.
yeah, I got the point that this S is the fixed value.
Yeah.
But still through the derivation, all the derivation,
Yeah.
they assume that this fixed value as the initial condition
Yeah,
is
yeah.
somewhat preserved throughout the whole time.
Mm.
So if that is not the case, then we cannot simply say that like W sub ES cat and W sub ES, what was that? Cat and off or something.
Mm.
They would not have the same distribution, even after we assume that they are all exponentially distributed.
Mm.
Because the number S literally goes to S minus 1.
Yeah.
If W cat happens, but if W off happens,
Mm.
then S is still S.
Yeah.
So that can still change. So
Mm. Yes.
okay.
I should check the derivation later. So
If you really want to
yeah.
use
Yeah.
some equations from this paper,
Yeah.
then we have to keep in mind.
Okay. But I'm not sure whether they actually mentioned that S is changes or not.
Yeah.
In the
Yeah.
derivations. So
Many authors don't.
Can you go back to the page 10?
10?
There's a quote that you proposed.
10? Page 10?
The right below the B, the S goes in and out in the restricted local area of the enzyme.
Mhm.
So I assume that if this picture is used by the author, they assume that S can be changed.
But actually, this pictures comes from another paper. I just bring this paper as a sort of these pictures was understanding of this thing of Michaelis-Menten.
If that is the common sense in this area of the field,
Yeah.
you can assume that S and P and all the variables are changing in the experiment.
Yeah, right. But I'm not sure whether this is in the equilibrium so that the potential changes does not occur or not. It depends on whether this is in the equilibrium or not. In an equilibrium.
If you have like single enzyme, like huge pot, and then you know that there are like tons of substrate and products, and they are all well mixed. And then the region of interest is kind of large, so that there are always more than hundreds substrate who could interact with the single enzyme. Then everything is well approximated. But if there are only 3 substrates who can interact with the single enzyme, then although the other substrate can go in the region of interest, and then they somewhat preserve the level, but there will be a small time window that actually matters. So
Right, it's just my impression of this paper. They really like imagine the situation where the typical in vitro experiment, like where there is high substrate concentrations of substrate condition,
Yeah.
really doesn't change. But I think I'm not sure about this paper like aims those kind of situations.
Yeah, I also think to fit is actually I also think like that maybe this model assumption may be quite fit to the in vitro experiments. So maybe I should be more careful for reading other type of these papers. Other types of these papers. So any other questions? Okay, then let's go to the non-Markovian enzymes. So actually, they exist for the Markovian enzyme, but for the non-Markovian enzyme, hidden values are not obtainable even we use like 1,000th order of the moment. So let's say that this table again, and then let's check whether it is possible to obtain the hidden variables with the higher moments. So let's say we only use this omega-1, then since we can obtain the A1 and B1 values from the experiments, we have some linear equations, two linear equations, but the number of the hidden variables are 3. So the hidden variables is not obtainable. And if we use two omegas, omega-1 and omega-2, then we obtain the linear equations. We obtain 4 linear equations. Actually, no 4 linear equations. The 4 equations. And but the number of the hidden variables increased to 5. So we cannot get hidden variables from the experiment. And if we use until 3, then the number of equations increased to 6. But the number of the hidden variables increased to 7. So actually we cannot obtain hidden variables also in this case. And they mentioned that in general, if we use until nth omega, although we obtained 2m omega from the combination of the higher moments and from the experiments, actually this relationship contains 2m plus 1 hidden variables, so that we cannot get the hidden variables if we are dealing with non-Markovian enzyme. So this is the problem, so that we cannot obtain the hidden values completely without any additional information, such as if we know that, actually if we know something like the k-on values, then we can get the hidden variables from this relationship. But if we don't have any additional information, then we cannot get the hidden values completely. So what the process that they took was just simply assuming Markovian enzyme. Then hidden values can be obtained within the bounded error vectors. So here is the non-Markovian enzyme, the values of the k-on, pi cat, and the waiting times. And then in the non-Markovian enzyme cases, it's not obtainable. So they just simply assume Markovian. Then we can get each values. And then if you can see that if we use the estimated value of the lower case, and then you can see that it differs to the true values as factored by these values, where the mean of the waiting time of the ES over the mean of the catalysis of the ES. So it only differs like these values, like here, it product factored by these error vectors. Then what they saw is this error vector significant, so that we cannot take this, just simply take the Markovian assumptions. So they compute these values in a representative non-Markovian enzymes by just deriving these values within equations, and then they draw the heat map of these error vectors under the various parameter values. So they first focus on the sequential relation that I explained before. So it contains something like multiple steps for this complex. And if they draw the values of the error vectors, it looks like this. So here is the x values is parameters like this, and y value is the other parameters, and this color means these error vectors, which differs from the simple Markovian assumption and then the real non-Markovian cases. And as you can see, wide ranges of the parameter regions, these error factors only has values from 1 to 1 or 5. So that it means that although we assume this simple Markovian assumption, these estimated values only differs in maximally, not maximally, but in this range is maximally half from the true values. If we also focus on other cases like parallel reactions, where this enzyme substrate can go to this enzyme can bind to substrate 1 or substrate 2 within the probability P. It also take this error factor values. Here, error factor, the blue color means it close to 1, and then the right color means it goes over the 1. And then as you can see, in the wide ranges of the parameter values, the error factor is only close to 1, so that within these regions, this simple Markovian estimators only deviate the true values like controlled by this 1 or 2.5 or 5.
Oh, so
Yeah.
why they compare the WES over WES cat?
Actually, under the Markovian assumptions, this WCAT ES and then the ratio of the WCAT ES and the WES are 1. So, let's say that let's bring the K1 case. K1 case. K1 should be like this. And then this values is fixed by experiments. But we don't know these values. So they simply assume this, I would say this is gamma. This gamma is just 1. So that they can get K on from experiments. And then theoretically, the differences between K on tilde and K on is gamma, which is represented in the heat map color variable. So they want to say that this gamma actually not that far from the 1. So that we can just simply assume the Markovian enzyme, and then we can obtain these hidden variables under some acceptable bias. So although there is some bias, but we can get the hidden variables very precisely and then identifiable.
And then the number of equation was 1 larger than the number of data. So if we assume that, then now we can get consistent system.
Right. So as we did in the Markovian enzyme.
So for those two kind of the Markovian cases, we should reduce the system into like K on K off. Get
Right.
variable system to do this.
Right. So actually, we cannot obtain this like K cat 1 or K cat. No. Like this. How can I say this K off exact K off values or the exact values of this K off from the experiment? They only. Wait. All right, they just simply compress this scheme into the linear approach, like E plus S becomes ES and then P. So they only get the waiting time, information of the waiting time of this WES cat or WES. So they actually, so the induced error that only affects like first or second digits of the estimated values. So this assumption ensures some acceptable bias but identifiable inference of the hidden values even in a non-microbial enzymes. So let's fail the actual inference from these sequential reaction cases by using some numerical simulations. And then this is the result, like comparing the true values and then the inference values. And then as you can see, there are some biases that makes the inferred values deviate from the true values. But actually, they say that this is something like acceptable bias. And then in my thoughts, actually in the pharmacokinetics, like I think the difference within like 1.5 fold or 2-fold is acceptable. So maybe since the differences are within the 2-fold, so I think it is something like acceptable bias. So
what is the meaning of N?
N is the number of the enzyme for the simulation. So it's something like the number of the sample of this turnover distribution.
Oh.
Yeah.
Because even if we have only one enzyme,
we can do like can we repeat like 1,000 times, 10,000 times, or like this time, so to obtain the distributions.
I see. So it doesn't really matter if we have like only one enzyme or 1,000 of enzymes. What the N stands for the number of samples used to make the distribution.
Right.
Waiting time distribution.
Right. Like the turnover time distribution. So the all hidden kinetic parameters are obtained, and this is actually impossible in a conventional approach. So if we accept this bias, then we can actually get these parameters identifiable. So this is the final slide of these papers. And then in summary, the conventional approach using Michaelis-Menten equation can only infer the compressed values like Kk or then the Michaelis-Menten constant. So it is a limited information for analyzing and utilizing the enzyme reactions. So by utilizing the data from the single molecular experiments, and then using the equations describing the combination of the moments, they propose higher order Michaelis-Menten equations, and then this higher order provides an accurate inferences of the whole hidden variables completely for these microbial enzymes like this. So it serves some unbiased and identifiable estimations. And for the non-microbial cases, this approach is incomplete, but can be utilized by simply assuming the microbial enzymes. So error is still bounded so that we can get acceptably biased but identifiable estimations. So this is the references, and thank you for taking care of my presentation.